We, oops, sorry, I get that. It's always so disconcerting. Recording in progress, like, always kind of stresses me out every time that happens. Okay, so today joining us is Dr. Brian J. Bennett, who comes to us virtually from the Western Human Nutrition Research Center out at UC Davis, and he is the research leader in the obesity metabolism unit. He shares the same passion that I think many of us have, which is to help prevent heart disease. He researches the role of human diet and nutrition as it relates to metabolic disease. And in particular, he's interested in the genetic components of cardiovascular disease and obesity through ingestive genetic studies, also called systems genetics. I'm sure he's going to share some of that with us today. His lab examines the relationship among many types of data, such as genetic variants, gene expression levels, and metabolite levels, and how these interact to create susceptibility to cardiovascular disease. In particular, he's interested in this TMAO metabolite, which I'm very excited to hear more about, and he's going to share that with us. Just to give a little bit of background on his kind of where he's from. He did his bachelor's of science in exercise science at Ithaca College, beautiful campus, in 1995. He then got his master's degree in nutritional sciences at the University of New Hampshire under the guidance of Dr. Tony Taliaferro, one of my favorite scientists. So that's really exciting for me. Then he's done his PhD in nutritional sciences at the University of Washington. He did doc work at UCLA. And then he was an assistant professor of genetics at the University of North Carolina, Chapel Hill. So he's kind of been all over. He's been very well funded, and he's going to share with us today some of his current research. So really happy to have you. So welcome. And with that, I will hand over the Zoom stage to you, Dr. Bennett.
Thank you. That was a really nice introduction. I appreciate it. And I really appreciate being invited to come share a little bit about my research. And I am envious because I noticed that you guys may have these walking treadmill desks, which was actually, I just was totally blown away by that. So the idea if I could be actually getting some exercise while talking to you guys, that would just be amazing. So
it would, but I'm sitting here beside mine right now as we speak, so don't feel too badly.
Yes, it's so I'm going to get started, and this can be fairly informal. I know you guys do things in chat. If anyone feels compelled and needs to yell out loud and say, hey, stop, I need to understand this, feel free to just interrupt. I won't take offense at that. And I'm going to get started right now, if I can remember where this little guy is. There we go. So today I'm going to talk about genetic background shaping phenotypic response to diet in the collaborative cross. So it's a very mouse genetics talk. And so we can, just to fair warning for anyone that's not as familiar with that. So here's some of my affiliations here in Davis. And one of the things that I lead is this group on obesity and metabolism. So here's the metabolism maps. We see them from the CDC. A lot of people will go through the chronology of it. And one of the things that always strikes me is there hasn't been rampant genetic mutations, we think, that have happened in the last 50 years, but there's been this massive increase in obesity. So it's this kind of the susceptibility to a new environment that seems to be really, really something that we need to focus on. And so one of the things that I think about as a USDA scientist is that some of our mission and some of the things that we do here. And so the unit that I work with and supervise is focused on understanding the dietary guidelines. And so for many years, the USDA and Health and Human Services have put out a joint report on what are the dietary guidelines for Americans. And we study that in our nutrition programs. And more recently, they've tasked us with trying to design diets and provide interventions that help us understand how effective the current recommendations are. So hopefully we can refine them. And this is really, in the context of the map we just saw, is a little bit disconcerting because the map keeps on getting worse in spite of our recommendations. And so trying to understand where that interface between maybe policy is or how our research may affect policy later is one of the things that we keep in the back of our minds here. It won't be front and center in this talk, but we're hoping that we can understand some of these things a little bit better. And so this is the building a few years ago. And the way that we do that is through some intervention studies, and there's some really good intervention scientists here and some other folks in the center looking at eating behavior and other aspects of metabolism. So I won't go on in there too much, but to say that this is really our focus is to understand the dietary guidelines and how they're affected by or how humans respond to the dietary guidelines. So just acknowledging all of that before we launch into a talk about mice. And so what is the motivating study? And so here's a really, at this point, an old study looking at 4 different diet patterns or extreme diets. And it was an intervention study, behavioral intervention. And it showed no effect. There was really no difference in being on any of these diets. Was one of the things that was kind of surprising at the time. But I think about this, for some of these folks, so this is the absolute weight change at 12 months in kilograms. So some folks were over 30 pounds lighter. And then it didn't matter what diet there were some people that responded to that specific diet. And so that's an intriguing concept. So is it just that people can adhere to the diet, or is there something different about people that allow them to succeed on one pattern versus another? I think that remains to be a question. And so one thing that I do think about is, does adherence determine success, or does success determine adherence? And I just think that's an important thing. Many people are really going to lose interest with following a diet prescription if there's just no success. And in addition to the folks that were successful, there were folks that really was unsuccessful. They went the opposite direction with their diet intervention. So this kind of motivates some of our thoughts about inter-individual variation and the response to diet. So that's kind of what we're trying to do in some of these mouse studies. And so here's the crux. So, know that we're in Zoom times, and sometimes you're fighting with emails and other things that will go on in the background. Here's the crux. Disease susceptibility is driven by both genetic and environmental, in this case, dietary factors. And a hypothesis is that metabolic individuality affects disease risk and nutrient need. And the question is, how can we incorporate metabolic individuality or genetics into our nutrition studies? We can do genome-wide association, and that's a genetic approach, but it's way too big for doing like our classic feeding studies or really careful interventions. That's a population-based approach. You could do twin studies, you could do genotype guided interventions, and you can do studies in mice. And so the last one is what we're going to spend the rest of the talk on. And so this is something that I've been thinking about for a while, but clearly the NIH has really moved into this space tremendously in the last 12 to 18 months with their program on precision nutrition, and trying to understand how can we better inform the population of the US what they should eat. What is healthy nutrition? And so we're not alone at the WHNRC in our thoughts on this and interest in it, and I know a lot of other people are as well. And so here's a simple, quote unquote, a simple picture of what's going on here. And so this is just a reminder over here that we have diet and physical activity. There are environmental obesogens, stress, and probably many other environmental or behavioral factors in this box, genetics, and then things like microbiome. And they affect a lot of these things will affect gene expression, which affect hormone regulation, metabolism, and maybe susceptibility to obesity. And obviously this connection between obesity and other diseases is of primary interest for us. And so I'm going to break this up into 2 parts. One, just talking about the genetics, diet, obesity, and related traits, looking at genetics and diet, metabolism, and obesity traits. And then part 2, I'm just going to talk about gene expression and obesity. So both parts are going to be fairly heavy into genetics, and then some approaches that we use in systems genetics. I think in a way that hopefully makes sense to you. If we start to lose the audience, instead of hanging up, make sure that you say, hey, I don't understand, and we'll stop. If we don't get all the way, it's totally fine. Why mice for genetic studies? Obviously, you can tightly control the environment. You're given synthetically defined diets. You can regulate access to food. And most importantly, from a genetic standpoint, there are resistant mice and susceptible mice to many diseases or many phenotypes. And so one of the things I'm interested in is atherosclerosis. And that's something that does have some classic strains that are susceptible and resistant to that particular phenotype. Obesity is another one. And so there are hundreds of inbred strains of mice, probably well over 120. And early in the 90s, late 80s and 90s, a lot of people were doing strain surveys to understand which strains were susceptible to certain phenotypes. And so there's classic studies by Serwitt, and here's one that's shown looking at the effect of fat and sucrose on the development of obesity and diabetes in C57 black 6 mice or B6 mice, and then AJ mice. And what turns out is that AJ mice are fairly resistant to many diet-induced effects. So things like obesity and diabetes. And so this work, was seminal at the time. There were many follow-up studies. But most of our research for obesity is done in this one strain, C57 black. And so that's because they are susceptible to diet-induced obesity and then ultimately diabetes. So in my studies, I take a completely different approach. I look at many strains of mice. The two that we just talked about, AJ and black 6, are here. And then there are other strains such as 129, NOD, so that's non-obese diabetic, NZO, that's New Zealand obese, that's this really large mouse here. And then 3 wild-derived strains, one called Castaneus, one called PWK, one called WSB. These are challenging mice to work with because being wild-derived, they're small and super athletic. So there may have been a time where we've had to, wrangle 1 or 2 because they are very athletic. So I mention that because they're really phenotypically different. One of the reasons why they're phenotypically different is you can imagine when they're constructing these inbred strains over 100 years, at some point you pick a mouse out of a cage and you put it in another cage with the opposite sex, and you wind up making more mice. I've come to believe that perhaps the ones that were moved between cages were a little bit slower, just because you have a technician trying to catch mice. So these 3 that are wild-derived have not undergone that selection. They're just really quick. But why they're so different is their genetics is different. And this is a phylogenetic tree showing here that Cas, PWK, and these other strains are just really far apart. They're really, really different. And that difference in underlying genetics makes their phenotypes different. It also makes the genetics different. So you have this way to look at what are the differences between these strains. And the strains are actually 3 different subspecies of mouse, and that's depicted here. And we get really excited about that in mouse genetics. It may not be as exciting for you guys. In some of our early studies looking at these collaborative cross-founder strains, so these 8 strains I mentioned, their genetics are different, their phenotypes are different. And that makes a lot of sense because a mouse, if you just saw this mouse NZO and then you saw Cas, they'd be like 40 grams different in size. So, they'd be on the order of like a 600-pound human and a 90-pound human. So that's just really different. And so these studies were set up to understand which strains are susceptible, and then in particular, looking a little bit deeper at some of the metabolites and the connections between the microbiome and these phenotypes. But we also did a few things where we would manipulate the diet with these strains. And this is just an example showing that there is a tremendous amount of variation within each of these bars that I have. So the colors are the strains. And then this is a control diet, AIN 93. This is a high-fat cholic acid diet for this study. And so what we see also is that when we add the diet, we maintain this level of variation, or maybe accentuate the level of variation between strains for each of these phenotypes. And these are things like ALT, total cholesterol, triglycerides, and then one that didn't seem to change with diet was plasma TMAO, something I'm interested in. So some things change, not everything. And it does seem to be that they're strain-specific susceptibilities or increases in some of those phenotypes. So I've told you about these 8 strains of mice, the fact that they're really different from each other, and then that they do respond to diet. And there seems to be a little bit of a variation in how they respond to diet. So the collaborative cross is kind of the next step in this series of studies. So this is something that was dreamed up in the early 2000s, where they were going to make 1,000 recombinant inbred mice. And so recombinant inbred mice are strains of mice where you've kind of mixed the genetics in order to use it for genetic mapping and systems biology. And so this was a very forward-thinking approach in the early 2000s, and they started this project. And one of the reasons why I went to UNC is that was a site that had most of the strains that were available. And so just to understand what's going on, there's 8 strains of mice, and we want to mix the genetics for those 8 strains of mice. So we go ahead and we put males of one strain with females of a different strain together, and we make offspring. And then we go ahead and take those offspring, intercross them. And so what's shown here are there was this funnel approach in order to do the breeding very quickly in order to fix alleles, so that way you would have an inbred mouse. And so one thing that's important to remember is that for an animal to be inbred, you need 20 generations of breeding. So this funnel is like many postdocs' lives, right? It was a really large project that took years and years and years. And so at some point, there was a subset of the mice they started off with that were available, that were fully inbred. And these are some of the things that, some of the strains that I'm going to talk to you about. And so one of the things that is important here is that we can reproduce the studies since they're fully inbred. The data is kind of additive. So you can do a study at one time and add some mice to it later, or you have the same stable genetics to look at 2 perturbations. And so that's really fundamental. The other thing is that you can have multiple replicates, or in one way to think about it would be twins. So you can have multiple twins per perturbation. So if you want to feed one group one diet, one group the other diet, you can have replicates. And that's fairly important because our diet responses can be a little bit noisy depending on behaviors like, cage dominance and how much a particular mouse eats or doesn't eat. So here's another picture of that sort of thought. So, if we have human twins or we have these inbred strains of mice, we can do this. So you can imagine that we can segregate these twins, one on diet 1, one on diet 2. We're not going to do that in humans very often. There's a lot, that's a really hard thing to do. But in these strains, we can wind up having, CC strain 28. So that's one of the ways that they're known is by a collaborative cross, and then what line number are they? And then CC 12, and then say we're going to have multiple replicates of those strains on one diet versus another diet. So what we wind up doing with those mice, or what we did was we wound up looking at 2 factors. One was heritability. So what is the overall contribution of genetics to traits such as obesity during a phase where we're just giving a standard AIN diet? And then we perturb the diet by either giving a high protein diet or a high sucrose diet, and then look at what are the effects of diet. And so really kind of not a very complicated design, but what we really trying to do is understand what is the contribution of genetics, and then what happens when we perturb diet. And the advantage of this particular approach, I would argue, is that we have multiple replicates per genetic type. And these are kind of some questions. How much do genetics and diet contribute to the development of obesity? Does diet alter susceptibility to developing obesity? And do differences in micronutrient composition result in beneficial phenotypic outcomes. So we do things, and I'm sure that that you guys do similar things in your mouse studies. Often, if you're looking at energy balance, there are things like MRI approaches to look at adiposity, and then I think what is the percent body fat as opposed to just how much they weigh. And there are things like indirect calorimetry, which we started off talking about in pigs. They actually have these fancy systems that can look at how much they move around, do they rear, how much do they eat, and then also what is their RER and other indirect calorimetry measures. And so we put a bunch of these mice through this battery of tests. And what we wound up doing was we would get a cohort of 22 strains, 3 mice per strain, run them through, get another cohort of 3, run them through, another cohort of 3, in order to kind of replicate. So just describing a little bit more about the study. And so in this acclimation phase, this is what we saw, which was a tremendous variation in just body fat percentage. So just typing the mice. And so what you can see here, things like body fat percentage, somewhere between 5 and almost 25%, or total weight, somewhere between 20 grams and probably more like 10 grams. And so this is really, fundamentally the variation within the population really does mimic the extremes of the human population. And that's really what we're looking for. And so this is just a reminder, I think partially for me, that as we're collecting each of these phenotypes and looking at them, we are trying to understand how are they behaving in the context of what we know. And so what we do know is that body fat percentage should correlate somewhat with things like lean mass, total weight. And then looking at some of the consequences of obesity, we would want to understand some correlation with insulin or insulin resistance and other things like that. And so we do see that, with unperturbed mice. So, just kind of everybody eating the same diet. And so then we put them on not necessarily really extreme diets, a high protein diet, and then a high fat, high sucrose diet, really trying to manipulate the macronutrient component of the diets in order to understand what are the responses to changes in macronutrient composition. And so we feed them for about 8 weeks, 9 weeks, and then we wind up looking at body composition, doing our indirect calorimetry, and doing some further phenotyping. And so one of the things that you can notice right away is that certain phenotypes are going to respond to changes in macronutrient composition more than others. So things like body fat percentage really didn't reach the level of significance. So there wasn't, if you looked at the population, there was no difference between body fat percentage on either of these diets. And so that mimics kind of what we saw in the human study earlier, the motivating study. And then there are things like weight or triglycerides, or actually weight and cholesterol, which do seem to be a little bit more responsive to a high sucrose diet. So when the diet has more sucrose or less protein, because those things kind of travel together in this study, you wind up having increased cholesterol, increased total weight. So not necessarily unexpected results. This is where it gets kind of interesting, is that, so we've colored each of these phenotypes. So this is body fat percentage, total weight, triglycerides, total cholesterol. These are the 22 strains across the bottom. And then we have the diets colored. So pink is high protein and blue is high sucrose. And what you can see is that there's still this graded effect where there are fatter mice and skinnier mice. But what's really interesting is when you look at this and you realize that there are some strains that are non-responders. So CC63 doesn't really respond to macronutrient changes. But if you look at CC28, there's something about those mice that they're going to gain a lot more fat when they're fed a high sucrose diet. And conversely, CC12 look like they actually are losing weight or not, are non-responsive to this high sucrose diet. And that happens for body fat percentage. And you can also see it for other phenotypes. And it's not always the same strain that's affected by the same for different phenotypes. So these are ordered by the highest strain to the lowest strain. So I just want to point out the highest body fat percentage strain is CC40. The highest triglycerides is CC36. So they're different strains. They're not ordered exactly the same, except for some of these strains that are non-responders can wind up just being kind of like AJ, I guess, really non-diet responders. So this is really, again, one of the motivating study was try to understand, like, is it possible that overall there's not a diet effect, but there's an effect based on the underlying genetics? And this is really one of the things we've been trying to understand. So quite exciting for us to see this. The other thing that this model that I use allows for is the ability to calculate heritability and then calculate the effect size of the diet for an individual strain. So I'm going to show that next. It's a heat map. And since we can collect lots of phenotypes on these mice, that's what's shown here. The really thing that the important thing I want to contrast is that the heritability, so the amount of a trait that's regulated or associated with underlying genetics varies by the trait. So certain things really are not very heritable. So some of the insulin responses, fatty acids, they're more variable, and so it's not as consistent within a genotype. So and then other things such as lean mass, it is really does look quite heritable. And then when we look at the diet effects, it does look like the high sucrose diet had a much greater effect on the phenotypes that it does. That's a terrible sentence. So it's the high sucrose diet does seem to affect certain phenotypes to a greater extent than the high protein diet. And that's kind of shown by the darkness of the blue here, of the green, excuse me. So the effect size for certain things like total weight, lean mass, and things like unknown things like why would choline be particularly higher in the high sucrose diet is a little bit unclear. So this was, again, another fundamental, I guess, result from this study, trying to understand what is the balance between genetics and diet effects. And so here's just a summary of where we are. Genetic architecture influences obesity in the collaborative cross. The variation of strain is probably better than the effect. No, it's not better, it's greater than the effect of diet. Genetics largely determines whether an individual will become obese. The effect of diet can be larger or smaller depending on the specific underlying genetics. And I think that's fairly important. So a high sucrose diet was going to affect certain strains more than others, and a high protein diet can have effects in other strains. And that's, also kind of an important consideration as we think about dietary guidelines. So that's kind of the first paper that one of the grad students in the lab, Phoebe Yam, published last year. I'm going to move into unpublished work that she's just finishing her dissertation, and just wanted to share some of that with you guys, where we look at genetics, diet, and then we start to look a little bit more at the transcriptional landscape that's happening. And we focus on the liver. It's just a tissue that kind of makes sense when you're looking at obesity and cardiovascular disease, but other tissues such as adipose tissue or skeletal muscle would obviously make sense as well. I'm going to take a second to answer a question. So are we looking at sex as a biological variable in these CC mice? In this study, they're all females, but that is something we are really interested in. And we've been looking at that in an analogous population called the diversity outbred, but we haven't yet tackled that with the CC mice. Our next study is going to include males. So, and then I'm gonna stop with the chat for a second. Otherwise, there are such good questions. I got distracted, so I apologize for that. So I left this slide up for a couple of seconds. We're going to walk through genetics of gene expression. And so this part here was something that as a nutrition student, I didn't know existed until I got to do my postdoc down at UCLA with a mouse molecular geneticist named Jake Lucis. And so some of this I'm going to walk through, and I think we're going to hit the time just fine. So here we go. So what we wound up doing was we took out the livers from the mice, we flash froze them, we processed them, and then we did, this actually is microarray data, not RNA-seq data. And one of the reasons to do that is just the number of mice we were doing. It just wasn't feasible to do RNA-seq from a cost perspective. And what you can imagine is if you look at 23,000 transcripts, a number of them are going to be correlated with body fat percentage. That kind of makes sense. Here's a list of some of the ones that are most highly correlated. And as you would probably suspect, if you're correlated with body fat percentage, you're also going to be correlated perhaps with phenotypes that are correlated with body fat percentage, such as some of the metabolic signatures out of the indirect collimators or plasma chemistries for things that are related to adiposity. We also can take those 23,000 transcripts and say, well, which ones are different between the two diets? And so, that was, again, thousands of genes that were differentially expressed by diet, seemed to be fairly unique from the ones that were correlated with body fat percentage. And then this was the one that was kind of surprising. Nearly, not quite half of the genes were differentially expressed by strain. So just thousands and thousands of transcripts were different between the strains. And that, when we took a step back, that does make sense. If you have these CAST and PWK and WSB mice that are so genetically diverse in this population, so those alleles are segregating within the population, you're going to wind up having just differences in gene expression. And just to point this out, so these are all heat maps. And so on the bottom are the strains. It's probably hard to see. At the top are the colors representing a strain. And so the ones that are next to each other are this is the average of a strain on one diet. And then this winds up being the same strain on the other diet, is kind of how this is represented. So we were kind of happy that the strain effects seem to be a little bit stronger than the diet effects. So it's not like the diets are all over the place. It really is the strains are kind of next to each other. So that was kind of an interesting result. And so one of the things that we often do, and you'll see this often in papers, is just some ontology or some enrichments. And so I have this line here, kind of just dividing up things that are overrepresented in the transcripts that are affected by diet. So some metabolic pathways, and here's some of these other things such as amino acid catabolism. And if you overfeed or if you feed a high protein diet, you would think that might be different between the diets that are manipulating macronutrients. And then here's some differentially expressed genes by strain. These are a little bit harder to think about. So cell adhesion molecules, hematopoietic cell lines, regulation of locomotion, again, response to wound healing, things that are a little bit different. And they're probably just phenotypes that are different between the strains at a certain level. And this, I just want to point out, something like type 1 diabetes mellitus is actually a positive control in the study, because certain of the collaborative cross strains, because they contain genes from NOD, the non-obese diabetic mouse, having a difference by strain for type 1 diabetes mellitus is actually kind of makes sense. And so this is in this grand landscape of gene expressions, we can kind of segregate things into there. Some that are diet differentially expressed by diet, some that are differentially expressed by strain. So, and then how many are correlated with body fat percentage. And you can see that really there's about 350 that are responsive to diet, responsive to strain and correlated with body fat percentage. The strain differentially expressed genes, it's a larger number of those will overlap with the ones that are correlated with body fat percentage. So this is one way to get there. And so we kind of have gone through, there's an effective diet, there's an effective strain, and we're really kind of just focused on not everything that's different between these mice, but which are the ones that are correlated with body fat percentage. And so this is kind of a summary of what those things are here. And so we saw those results. We're really kind of encouraged. The next thing that we often do in some of these studies is something called make a transcriptional network. I'm going to walk through that in the last couple seconds here. So we use this thing called WGCNA, which is weighted gene co-expression network analysis. And what we're trying to do is take groups of genes and aggregate them together because their response is similar, and then try to relate that group of genes to phenotypes. Because we don't think that obesity is caused by a single gene. Most of it's caused by probably multiple genes that are interacting with the environment. And so that's kind of the approach that we took. And so there's a series of steps, and this is a really, a very mature pipeline for like bioinformatics at this point. Steve Horvath at UCLA and then Peter Langfelder developed this probably 10, 15 years ago. And here's just an example of the steps that you would go through where you make a co-expression network, and then you identify groups of genes or modules that are associated with phenotypes. And you really try to understand what does that group of genes do, or what does it enrich for? And so I'm just going to walk through how it works. So this is always a difficult thing. So you have a gene expression data set. And so that's represented here. So these are, what is the value, how much of this gene 1 is expressed in subject 1 or subject 2 or subject 2 or subject 3, and then this is gene 2. So you have this big flat data frame. And then you correlate every gene by every gene. And so obviously on the diagonal, you have a 1, and then you have your correlations. And so the thing that happens here bioinformatically is that we raise that to a power to create a scale-free network. And I'm just going to show what that does conceptually. So if you have two genes that are correlated with each other, one at 0.8, one at 0.2, if you raise the whole matrix to a power, you're going to diminish the correlation between everything in the matrix. But what happens are the low expressed genes wind up becoming much, much less prominent in their connection. And so these adjusted correlations that have been raised to a power then are transformed into an adjacency matrix, and you wind up looking at which things are most likely connected to something else based on the weight of this connection. That's a lot to think about. The most important part, I think, to take home is that this WGCNA is based on correlation, and what we're doing is trying to identify what are the core features or the core transcripts that are going to be correlated with each other and moving together as either diet or genetics is perturbed. And so this again shows the next step, which is then once we have the adjacency matrix, we wind up trying to say these genes or these transcripts are behaving similarly. So we're going to call them the red module. These are the green modules, so on and so forth. And so I'm going to walk into the next part. So we did that on these about 13,000 genes that we selected as being expressed in the liver. And so what we wind up seeing is that we have different size modules, and that kind of is a phenomenon of this particular approach. And they range somewhere between 42 genes and about 3,000 genes. And then we have here our, the first one is, again, what are the genes that are correlated with body fat? The second one is what are the percentage of genes that are affected by diet, differentially affected by diet, and then what are the percentage of genes in the module that are affected by strain. And so we can see that there's strain does have a very prominent effect in this approach. So something to take into account is that underlying genetics is going to drive how these groups of genes move up or down within a population. So we take this group of genes and then we correlate with all these phenotypes. And so this is a tough one to see on your screen, but basically what we have are, we've grouped the genes into all these modules that are listed here. And then we correlate the first principal component of that module with all of these traits. And really what we're focusing on still is body fat percentage. And so there are a couple of modules that are correlated with body fat percentage. And it looks like it can be positively correlated or negatively correlated at a certain threshold of significance. And so there are things like the magenta module or the yellow module are just two modules that we kind of got interested in the study. And so we did functional enrichment earlier, saying, hey, these differentially expressed genes are enriched for this, and and these differentially expressed by strain genes are enriched for this. We can do the same thing with the modules and say, what about these 42 genes is interesting, or what about these 100 genes is interesting? And so here we just show that endoplasmic reticulum is overly abundant in the magenta module. In the yellow module, circadian rhythm is overly abundant. So two things that kind of make sense about adiposity or pathways that are related to adiposity. Obviously, the peroxisome and other things are prominent as well. So we were just to show two examples of what we do with these modules. Is this ME yellow that is the principal component of the module. And so what we wind up trying to understand is, what is the effect of diet on the expression of the module itself? And so what we see is that there's a fairly large effect for the yellow module of it. You're going to have increased expression if you have if you're on the high protein diet. If you're on the high sucrose diet, the high sucrose diet are going to have a higher magenta module. So again, looking not so much at what individual genes are affected, but I'm trying to understand how a pathway or a group of genes is affected. And so I know we're closing in on time, and what I wanted to do is just acknowledge that also genetics is a tremendous driver here. So these are the 22 strains are listed down below, and what you see is that for a particular set of genes, there'll be certain strains will be high expressors, some will be low expressors. And so we're just, again, trying to really decompose the genetic and the diet effects into a way that we can come up with testable hypotheses. So here's kind of a summary. So we identify three main groups of genes or modules that were affected, that were associated with adiposity, and we kind of really dug a little bit deeper into those. We didn't get enough time to share all of that with you, but really what remains is what's important. And so we want to identify key mediators of a module response, and then understand if we perturb these mediators, can we affect adiposity? And so that's really the important thing, is can we find something novel, either a new transcript or a new transcript environment interaction that lead to increased adiposity? So that's kind of the crux of where we're headed with that. And so just some take-home message, genetics largely determines whether an individual become obese. And by individual, I mean a collaborative cross mouse, so maybe not a human. And the effect of diet can be larger or smaller depending on specific genetics. And that was really the first part of the talk. And then diet influences hepatic gene expression globally, but again, genetics defines the extent of the dietary effects. There really is this gene diet interaction that we're seeing. And we're really hopefully going to get to some testable hypotheses and, reopening the center in mass in order to do those studies soon. So I showed this slide earlier. I'm just going to wrap up for 1 second to let you guys know this is all stuff we did. Where are we kind of headed? And so we have this project that we work on testing the dietary guidelines. And so this is an example of what that study will look like. We're not recruiting for it right now based on COVID restrictions, but we are testing the dietary guidelines in energy balance and energy restriction, and then comparing that to a typical American diet in an energy restricted state. And really what we're trying to understand is one of the important dietary guidelines that came out is, achieve and maintain a healthy weight. And so in our initial studies that we did here in the center, it looked like there were some effects of having a dietary guideline pattern in energy balance in folks that were metabolically challenged. They were slightly obese and not quite diabetic yet. So, one of the questions is, well, what happens when you start to lose weight? And so that's what this study is. And then one of the things that I've added is this mouse component. And this is one of the things that when we're talking about understanding the effect of sex as a biological variable, we have these a study that will use 600 mice. Same sort of thing. We'll have 2 weeks to kind of let them acclimate to where they're going to be and what they're doing. And then we're going to induce obesity with this high sucrose diet, because it does seem in general to make most of the mice slightly more obese. And then randomize them onto a diet that matches the human diets that I just mentioned. And our real idea here isn't necessarily to prove that mice are the same or different than humans. It's really to understand, can we use these network approaches to identify things that we should look for in our responders and our non-responders from the human study. And with that, I'm going to stop so I don't go on and on for time. I just, wanted to acknowledge the lab. This is a pre-COVID shot of the lab over on the right behind you guys. Let me see if I can get there. This is Phoebe. She did all the collaborative crosswork and analysis. She's a student who just graduated from the Integrative Genetics and Genomics program in Davis. And then a lot of the other stuff that was done either at UNC while I was there, or there were a couple other people such as Eric Gertz and Excel Q, who wound up really helping out with different aspects of that study. And so with that, I'll stop and maybe take some questions and go from there.
Great, thank you. Why don't we all unmute and clap for our speaker? That was great.
Great, thank you.
Thank you for that. And there are many, many questions in the chat. So I can go ahead and lead that unless you would rather, Brian, go through those yourself, but I can kind of dictate those. We'll kind of go with it that way and we'll see how that works. I think you already in your talk addressed the sex as a biological variable a little bit. Next question is from Japna. It was really interesting to see the high sucrose diet having a higher heritability effect size for lean mass compared to high protein diet. I would have assumed the opposite. You want to speak to that?
Yeah, and so that was actually,
you already spoke to that one or not, actually.
I think one of the things is, and I was moving so fast at that point. So for heritability, we looked at it on the AIN diet because we didn't want a diet perturbation. We looked at effect size. And so what we saw was that there was a larger effect size on lean mass for the high sucrose diet. What that may really be is that the mice are getting fatter on the high sucrose diet, right? So when we're partitioning all this into like, lean mass and fat mass, I don't necessarily know that we can look at them separately. Does that make sense? It's within the context of their gaining weight. So
Right. Yeah. Thank you. That does make sense.
Okay, next question is, how do you control for microbiome drift between the various collaborative cross strains? Presumably, since you're interbreeding for 20 generations, there's potential for the microbiome within strains to drift over that length of time. And as that can contribute towards the development of obesity, is there some way you control inherently for that drift in your studies?
Yeah, so Ryan, that's a really good question. And so ironically, there's a slide I took out that we're on right now, because I didn't want to get into like a really maybe distracting part of the study. And what we do is, so when you're doing mouse studies, cage and microbiome, in these studies, cage and strain are generally 100% confounded. And so then your cage effects are confounded with your microbiome effects. So your cage effects of the microbiome are now confounded with strain. And so one of the things we did is we randomized the mice. And it's why we use female mice in particular. It's because if we start randomizing males at 12 weeks of age, we were worried about fighting and some other issues that happen. And so we actually had cages of mice with one white mouse, one agouti, one black mouse. So you can imagine just randomizing the mice in order to break the connection between cage and strain and microbiome. So we wanted to do that. I think the other part of your study is when you're inbreeding, you're not worried about the microbiome as much. What we've noticed is that we have to be very careful when we purchase the mice. So these mice are available from the Jackson Lab or UNC. You can purchase them as a set. And when they come to us, we try to do one round of breeding if we're going to do a microbiome study. And that's fairly important because over time, the microbiome most likely is going to change from the vivarium. So people are studying the effects of the microbiome in the collaborative cross. And so there's Federico Ray, who's at Wisconsin, has done a fair amount of that work. And it's actually pretty intriguing. Again, strain and microbiome are 100% confounded at a certain level, unless you're going to do a transplant. What he does is he finds those bugs and finds that cool stuff and then puts it through his notobiotic core to, show causality. Did I answer your question?
Seemed like a thorough answer.
Okay.
So the next question is about estrogen. My lab is very interested in estrogen, and my graduate student asks about estrus control in your females. So it's interesting that you chose females because they're nicer. I get that. They're not going to fight each other. But most studies, as you know, choose males. And so now there's emergence of research on sex differences in the role of estrogen. So what do you do about that? Are you just using all females? So you just kind of...
We're using all females, and we haven't, part of it is you can only, there's only so many things that you can control for and still have an experiment. So we didn't necessarily control for the estrus cycle. And I'm trying to think, because it's a great question. I don't know if anybody's looked at differences within the collaborative cross, because you would assume there could be small variations between the inbred strains. The other thing is that because I'm so interested in atherosclerosis, our initial studies were in females because they're more susceptible to atherosclerosis. At least mouse models are more susceptible. So that was one of the reasons to do that.
Interesting. Yeah, in my experience, doing estrus cycling is way more work than it's worth, and it probably stresses the animals out more than it's worth. So it's one of those things that's hard to get around. Since the non-responding mouse strains are virtually unaffected by macronutrient changes, would you say that they would be affected by overall caloric intake instead? In addition, regarding the greater effect of high sucrose versus high protein, could that be related to inflammation caused by consistently higher sugar intake over time?
Okay, so looking at that question, we didn't test whether overfeeding or underfeeding the mice would have an effect. I think it's a separate question. It's a good question, which is, when would that work as well? Or would those mice that are always 15 grams, would they always be 15 grams? Would they find a way to survive? So I think that's another good study, but not one that we can maybe answer here. And the other thing would be that inflammation, we haven't looked at, like plasma cytokines in these mice. There are inflammatory pathways that are upregulated in the liver fairly consistently. Looking at, innate immune signaling, it's probably Kupffer cells and some other things like that. So is there systemic inflammation? Probably, but we don't have a blood measure of that.
Got it. Next question is about pathways associated with Parkinson's disease and Alzheimer's disease. And I think you had some data in there maybe to suggest that diet seems to play some role in affecting those pathways. Can you elaborate on any of that?
Right. So that's, you guys were paying attention really well. So thank you. When we're doing gene ontology, it's always important, at least I remind myself, that what we do is we have it's a data-driven approach. So we have our data and it's behaving a certain way. And then we go ahead and we interrogate a database and say, this unknown data, what does it have in it? And so when we use the collaborative cross, we're seeing some representation of Parkinson's disease, Alzheimer's disease. It may be that there's underlying genetic susceptibility to those diseases in the mice, and it's not a diet effect. I think that's important to recognize. And that's, so I don't necessarily know that we have any strong, compelling data to say either of those diets affected Parkinson's disease or Alzheimer's, because we didn't test the kind of the biochemical or the pathological, if we had brain sections for Alzheimer's or something like that, or had phenotyped them from Parkinson's, we might be able to stretch it, but not quite there yet.
It's complicated, lots of interactions. Next question is sort of a methods question. Would qPCR experiments to look at mRNA be redundant to perform on this type of study? That's more of a, sometimes we use RT-PCR to validate some of these big changes that you see. Have you done any?
So that's, it's a great
targeted analyses.
Yeah, it's a great question. And so we've done some of that sort of work. I don't know if we've done any qPCR in this study. In general, the things that we find by the microarray, there's such large effect when we're looking at the diet effect or the or the strain effects, that things tend to be consistent in these kind of systems genetic approaches. Doesn't mean that there's no value in it. The one thing that I think there is some value is if we were looking at a splice variant or some other specific variant. So let's say I landed on a gene that I'm interested in the lab is CD44. So if I wanted to look at CD44 specifically, I may want to do some sort of higher resolution approach than just the microarray. I think the extension of your question is, if I had done RNA-seq, should I do qPCR as well, right? And that often is something that folks will get asked in review, and it after you've done RNA-seq, it can be a little bit like, well, I think I've done enough, but I'll do it because you want me to.
Yeah, sometimes it's never enough for the reviewers,
yeah.
Right? Next question is, do you perform WGCNA on each time point of the cell intervention separately? That's Jabna's question.
Yeah, and that's, one of the things, and this is one of the things that would have made the study maybe better, would be if I had kept mice on the AIN76 diet all the way through. So I don't have, in a way, because when we're taking doing the transcriptional network, that's a terminal thing. So, we're pulling the liver out and, doing all the microarray stuff. It would have been an advantage to have the AIN76 diet to look at, because we're also getting an effective age over the study. And so, so that would be one thing. So we don't do on the diet interventions, the time point separately. And we went through the WGCNA stuff kind of exhaustively. And so we landed on trying to make one network and decompose the diet and strain effects.
Cool.
Yeah, thank you. It's because I perform some of these analyses myself, but I'm a clinical scientist, and so I run clinical trials and try to see the longitudinal effects and change over time. And sometimes just modeling that when you have so many different time points in the same network can be very hard, and it just wouldn't make sense. But I like what you presented, and I'm going to go back and rewatch this seminar just to figure out how to do that.
Sometimes it's nice to have these recordings.
Yeah.
Well, one thing that I think is important for the WGCNA that we're doing is because we can use genetics as a causal anchor. And I think the time course stuff is a little bit more complicated. You do have inferred causality because you have a perturbation here and then an effect here, but it gets hard to model. I agree with that. And, most of the stuff that's network based is all coming out of, behavioral sciences and things that are much harder to do than some of the mouse work. So yeah, there's interesting stuff.
Great. Well, that was perfectly timed. It is 5 o'clock, right on the nose. It's too bad you're not here because we would go right to happy hour at this point. You need discussion, but you're not. So we'll end it there. Thank you so much, Dr. Bennett, for being here. This was a fantastic seminar. And I assume that you're okay if students have questions and would like to email you, or if anyone has further questions about your work, we can shoot you an email and
please do. That would be great. I would be more than happy to do that. And I think you guys have my email, so you can pass that to the students.
Perfect. Well, we all really enjoyed this. So thank you so much for your time.
Thank you.
Thank you so
All
much.
right.
Bye.
Take care, y'all. Bye.